Okay. So I'm Laura Bryant. I'm from the Merowitz Center, and welcome to today's intriguing program called Sewer Science. I'm happy to introduce Dr. Marc Johnson, a professor of molecular microbiology and immunology at the University of Missouri-Columbia. He's been studying this field for many years, and his lab often handles more than 100 wastewater samples each week from all over the country. Since COVID appeared, however, Dr. Johnson's cutting-edge work has made him a bit of a media celebrity and a go-to subject matter expert when it comes to wastewater surveillance. As a result, we really appreciate that Dr. Johnson was able to find the time to talk to the Merowitz community, and we look forward to hearing more about his research and findings. So I'm going to turn this over to you, Dr. Johnson, and thank you again for being here.
Sure. Can you all hear me? See my screen? We good? Okay. Let's see. So let me start by telling you a little bit of my presentation. I'm going to tell you about there'll be three parts to the presentation. The first will be about detection and quantification. So this is just our early days of SARS-CoV-2, where we realized, oh my God, we can study the virus from wastewater, which was kind of cool, and where we went with that. The second part will be about classification and traffic, and when we realized, oh, we can take this a step further, and we can actually figure out what viruses are circulating using this technology. And then if you stay with me, the third part is a little bit, gets kind of interesting. It's what was my top secret research about how wastewater can be an oracle, and how I became a published poet. And I just need to warn you now, I can't help myself. There will be some shit jokes. It's just, I try and stop it, but it just keeps coming. You know, it's just the puns are endless, but I hope I don't offend anyone. But there will be a few poopy jokes in here. So this all started in the beginning of 2020 when the COVID had first hit. There's actually some labs in the Netherlands and a few other places that first reported they could recover and measure SARS-CoV-2 levels from looking at just poop, looking at wastewater. And this was very quickly becoming an interesting thing. And I had nothing to do with it. I was happily in my lab studying HIV. No intention of becoming a wastewater epidemiologist, but it started with an email from a gentleman at the Department of Health and Senior Services at Missouri. They wanted to set something like this up in Missouri, and they didn't have the capacity at the state lab. And so they were just reaching out to the university, whether there was anyone who knew how to do PCR. And somehow they got my name, and I couldn't find anyone else that would do it. So I eventually got roped into saying, okay, I'll give it a try, but I didn't do it by myself. So I have a counterpart in all of our work, which is Dr. Chung Ho Lin, who is from the Department of Natural Resources. So we have a division of labor. I know about viruses and sequences, and he knows everything else in the universe. This guy is amazing. I mean, I can give him a poop sample, and he can tell me whether I was in a good mood or not. He can do anything with these. He's a chemist. He can learn anything with a water sample. It's incredible. I'll give you some examples in the talk. But overall, though, this is a partnership with the state. It was actually initiated by the Department of Natural Resources, who's the one who first started this. They're the ones that have relationships already with all of the wastewater treatment facilities, and realized that this is something we could do efficiently in Missouri because we already have this network of how wastewater is sent to a central lab for testing. Here at the University of Missouri, we do all of the testing for them. So all of their shit comes to my back door, and I take it and do the analysis, and then we quickly turn around the data and we give that to the Department of Health, and they're responsible for all of the communications. They manage the website, all of that stuff. And then once we got going, a lot of other groups jumped on board. So we're also regular partners with, we test every prison in the state every week. Also Department of Mental Health and Department of Veterans Affairs, where so there's a lot of focus on vulnerable populations. So we get these are the sites. This slide is constantly out of date because the sites where we do sampling keep changing. But these were the sites at one point where we were sampling all the wastewater treatment plants. It looks like we don't really have much of the state, but if you notice, the places where we don't have much sampling is just because there's no one who lives there, or not no one, but you have to have a wastewater treatment plant for us to study. And so We haven't started going door to door asking for samples from septic systems, but that might be coming, who knows? But at the moment, these are the places that it's between 60 and 70% of the population of the state's poop passes through my lab every week. So how does it work? It's not as gross as it sounds. When samples arrive in the lab, this is how they look. We get about a quarter cup. What we do is we first spin out the solids, and I assume you all know what I mean by solids. We then filter it to remove anything larger than a virus, so remove all the bacteria, and we then concentrate it by centrifugation, and we extract the RNA. And at this point, what we're doing is exactly the same as what happens anytime you've had a nasal swab taken, which is sent in for PCR testing. It's exactly the same technique. In fact, one of my former students was doing all of the COVID testing for the state health lab when we were getting started. So I just called her up and I copied all of their techniques. So we're doing exactly the same stuff that the state health labs are. The only difference is that when we measure RNA, when we measure how much viral material, we're not only asking, is it there or not? We're saying how much is there, so we can actually report back whether it's going up or down in various sewer systems. So we haven't seen it. We have a dashboard. You can go on and you can go to any one of these cities and you can click on it and you can see, where are we? Are numbers going up? Are they going down? You can look at where we were from the beginning of the pandemic to put everything in perspective. No hiding it. We get samples from everyone. It doesn't matter whether they got tested. It doesn't matter whether they have health insurance. It doesn't even matter if they believe the virus exists. As long as they're pooping and they use a community wastewater system, we'll find out. So one of the things the health department did early on is they really wanted to be able to see whether to test whether this was helping, whether they really were to testing things that reflected the actual patient infection. So what they did is they got detailed maps of all of the sewer sheds we were looking at. So like down to the household, we know exactly whose poop is going into the sewer sheds we're testing. And whenever someone tests positive in the state, particularly during the pandemic, you get their home address and it pings our facility that we know exactly how many cases have been diagnosed from each sewer shed. So we can do really detailed comparisons. And this is my first graph. I'm not going to go in the weeds too often, but this is just one. So this is early on. What we did is we took every one of these dots is a wastewater treatment facility. And what we did is how high the dot is reflects how much RNA we detected cumulatively over about a 2-month period. And on to the left and right is how many how many patients were diagnosed in that sewer shed. And you can see it makes a nearly straight line. The more patients there are, the more RNA we see. But you'll see that there were a few outliers. And this is something we spent a lot of time trying to figure out in the first few months. Is like when you have something that has more RNA than you would expect, does that mean that they weren't testing enough and that there's actually more patients there than they think, or is something else going on? And so I'm going to give you a few examples of places where it wasn't lack of testing, but there was a perfectly good explanation for why the numbers are off. I don't know if you know, this is the city of Charleston. Very early in the pandemic, they have a population of 5,000, and they had this huge outbreak in their amount of RNA. I was freaked out. It looked like 10 to 20% of the population had to be coming down with COVID. But we would look at their numbers and they only had a handful of cases. I couldn't figure out what was going on. Well, after a little time, we realized that we weren't really remembering that at the southwest of the town was the Southeast Correctional Center, which is a prison that housed 3,000 people, and we're experiencing a really bad outbreak. But the kicker was I forgot that the prisoners weren't registered as being their home address as being Charleston. So we're like, oh, okay, that explains that. We were actually, interestingly, we were even doing sewer shed sampling from that prison, and we're monitoring the outbreak. I just hadn't put it together that it was all the same sewer shed. And then there was another example, which was Kansas City. Kansas City's, their numbers didn't make any sense. They would tell us their flow rate, so we knew how much sewage they had, and we knew how many people there were. And the gallons of sewage per person were just ridiculous. It was like 400 gallons per person per day. And then we would do the numbers on the COVID, and they would have like 2 or 3 times more cases than we thought based on the RNA. And it would persist week after week. And so after a while, we reached out to the sewer shed, and we're like, you gave us this map of your sewer shed. Is this really the whole sewer shed? And they're like, yep, that's it. And we go back, and it happened. And after a while, we went back again, like, are you sure that there isn't something else? And they're like, oh yeah, did we forget to tell you? The Kansas side of Kansas City, their sewer shed is broken down. So they're sending like whatever 20 million gallons of wastewater to us every day. Did we forget to mention that? So our numbers were completely off because we were measuring all of these Kansas COVID numbers, but we didn't know about their cases. So that explained that one. But we had lots of examples of this over the first year. This is what kind of kept us busy. I just wanted to tell you about one other thing is like, what do we do? I've had this question so many times since you guys are mostly in St. Louis. I hear you got a little bit of rain lately. I got so many reporters calling me. It's like, what are we going to do with your sewer shed numbers with all of this rain? Because if you didn't know, in most of the St. Louis sewer sheds, when it rains badly enough, the sewers overflow and they just go in the river. And, most of the time we get, they tell us their flow rate, which is the total amount of wastewater they have in a day. But when they flow in the river, they have no idea how much went into the river. So we started working on an alternative to like figuring out how concentrated sewage is for correcting for this. We started this about a year ago, and this is again Chung Ho, who can do anything. We played around with a whole bunch of different things. So we were looking for markers, something that would tell us how much shit is in our wastewater. And turns out one of the most reliable markers of human activity is something we drink every day, which is coffee or soda or whatever, anything that produces this molecule caffeine, which, as it turns out, I'll measure the SARS-CoV-2. Chung Ho can actually put it into his little machines and figure out exactly how much caffeine is in our wastewater. And plotted out, it works. It's a remarkable reflection of how many people are in a sewer shed. And so this is our readout. So we do caffeine measurements every week. And so if the caffeine gets diluted, we know the sewer shed is diluted as well. But caffeine is just one example. As you can imagine, there are lots of small molecules that might be of interest. So Chung Ho can measure every opioid that is being secreted in a sewer shed. So it could be really useful. So like if there's an opioid problem in a region and you have a plan of how to fix it, we can give you an unbiased readout of whether the opioid usage is changing in a community. And it's completely non-invasive. It's just giving you a bird's eye view of what's going on. So now I want to move into part 2. So sequencing and characterization. So this was more my ballpark. And now I'm going to go into a little detail about what we do, but I'll get back to fluffy stuff if you're not digging it. But this is SARS-CoV-2. You've all seen this picture. So this little red knob you see on the surface, this is a protein called Spike. And what we have here is just a cartoon of the genome. And this green box is the Spike gene. So this red bulb has 2 jobs. It does 2 things for the virus. First is it binds to your host, what's called the host receptor. It's the protein on your surface that tells it what cell it wants to infect. And then it's responsible for penetrating that cell, of getting the virus into the cell. So it's really important for the infection. The virus is dead without it. So I'm going to blow this up. So this is what the spike protein actually looks like. If you really look up close. And so what you see is the way this is depicted is green is the thing that is on our cells that it's binding to. And this part of spike, which is actually binding it, is called the receptor binding domain. And what we actually do is we sequence that area. So this is nothing but a string of amino acids. So it's about 150 amino acids. This is what it looks like. It's just a code of proteins. This is the sequence of it. So what do we look for? One thing I need to tell you about is when you get vaccinated or when you've been infected, we produce antibodies. But what do we want them to do is the antibodies they'll bind to the spike protein, and especially if it binds to right at the receptor binding domain, that virus can't bind to its receptor anymore, so it's dead. It cannot produce an infection. And if this happens, if a virus is inactivated by an antibody, then we call this a neutralizing antibody. So that's good. This is what this is what you want to get from your vaccines. And if you remember, like if you've heard of monoclonal therapy against the virus, like Regeneron, the one that Trump got, and Eli Lilly has one as well. Sotrovimab, they've got weird names, but well, for a while, these were really good treatments for the virus. Well, that's what they are, is they're just giving you a whole bunch of these neutralizing antibodies that they've purified. So that's what a neutralizing antibody is. And that's really important because that's really our first line of defense against this virus. But here's what happens is that the virus will occasionally pick up mutations just randomly, but sometimes those mutations give it an advantage. So here's an example of a very common mutation that appears in SARS-CoV-2. It's called E484K. If for what it's worth, this was a mutation that was in both beta and gamma variants of concern. And what happens is if the virus has that mutation, a whole bunch of the antibodies that bound to the receptor binding domain don't work anymore. So they become non-neutralizing. So if you have a mutation that does this, that makes the virus resistant to an antibody, you call this an escape mutation. And what's important is that the main place where neutralization occurs is in this one little piece of the virus. So this is the piece of the virus that's picking up lots of mutations. And that's why this is the region we focus on for sequencing. Okay, so this is just my cheat sheet. All of our variants of concern, alpha, beta, gamma, delta, and all of the different Omicrons, pretty much all of them have different little fingerprints of mutations that they have in the receptor binding domain. So we sequence this whole area every week, but our output is just, I have a program that tells me what changes there are, and then I can match it up to our little key, and I can say, oh, we got these two mutations, oh, that's alpha, oh, these two mutations, that's delta, and that's how we figure out what lineages are in your sewer shed. So got it up and running, it was working, and that's when we started reporting variants by sewer shed in our state dashboard. And I'm just going to run you through sort of the last year and a half of what we've been seeing. So when we first started was the beginning of 2021. So this is right when we started hearing about variants of concern. And this green cross, so this first time we sequenced we could barely see any variants of concern. It was mostly just the original Wuhan strain, but there was this one this one green cross, which was the first time we had detected B117, which at the time was known as the UK variant and is now called Alpha. So the first place we saw it was in Hannibal and St. Louis were where we saw it in the very beginning. But within a few weeks, it started to spread, and we could see it spreading from sewer shed to sewer shed from week to week, and it was spreading in a very predictable way. So we saw it sort of work its way down the interstates, following kind of the population centers, and then over time, it eventually fanned out and went to the rural parts of the state. So this was in March, just to give you an idea of the information you get from sewer shed surveillance as opposed to patient sequencing. So the day before we posted this map, I looked up on what was the CDC website at the time. Although we were seeing the UK variant in 20 different sewer sheds, at that point, we only knew about 5 actual cases in the state. So as typical, the patient sequences are usually way behind. It takes time to get the samples, get the sequence, verify them before you know from sequencing. Whereas sewer shed, you just sequence it and there's your answer. So by the end of April, this is what it looked like. So it was mostly all alpha variant. These little yellow triangles means that there was more than one variant present, and almost all of these were a combination of alpha. And then we saw, if you remember the California variant, we had a fair bit of that around. Pretty much there's only 2 variants of concern that I've never seen, but most of them I didn't see. So I saw beta and gamma a couple of times. I saw epsilon quite a bit. That was the California variant, but mostly at this point in time, we were just seeing the alpha variant. But this, if you recall, this is the spring of last year. This is the first time we thought the pandemic was over. If you recall, numbers were way low. We barely had any infections. We were doing sewer shed, and a lot of the places were sort of undetectable. We thought, okay, how much longer are we going to be doing this? It seemed like things were wrapping up. And then we got an interesting readout. So this was the beginning of May 10th, in fact, where we got a new lineage showing up in Branson. And so this was, at the time, it was nothing. It was a B.1.617.2. it was not a variant of concern. Although the World Health Organization had said that this is probably the most contagious of the India variants. But at this point in time, it had not really done anything in the US. This is what turned out to be what was later named the Delta variant. So this was May 10th. We saw it only in Branson. And then the week later, already 1 week later, we were starting to see it around the state. So first thing is Branson has 2 sewer sheds. By the second week, it was pretty much dominated both of their sewer sheds. And then we started seeing it pop up in random places across the state. It was weird. There was this outbreak in Brookfield, Lynn County. I don't know if you guys remember this, but in May of last year, this was basically ground zero of the Delta outbreak. It was hell in Brookfield. They had like 3,000 cases per 100,000. Like 4% of their population was infected over about a 2-week period. Their hospitals were overwhelmed. And we got the readout, it was all Delta. We also saw it in Licking. It's like weird. It wasn't following population centers. It was just popping up randomly. And so this is 2 weeks after we detected the first time. It was already all over the state, but mostly in rural parts. So my assumption is that this was probably a Branson-derived illness. So I think these were probably people who had taken weekend trips to Branson and were coming home with a little present. Eventually, it did make it into the cities and the urban areas, but like Sedalia, Columbia is the last place where Delta arrived in Missouri. So then eventually the map looked like this. So turquoise square means Delta. And from June until December, this is what the map looked like. It was pretty boring. At this point in time, all the experts were saying from now on, all SARS-CoV-2 lineages will be Delta or Delta derivatives. And that was the commonly held belief. But we kept monitoring nonetheless. A few things interesting that came out of Delta is that we found that, after we had seen Delta arrive and move around a couple times, we realized that there was some predictive value. So this is just picking a few random sewer sheds where these yellow circles were the first week where we detected the Delta lineage. And what happened, without exception, is the Delta variant would arrive, and within 2 or 3 weeks, there would be a giant spike in their RNA levels. And that always was corresponding to patients. So this is when we actually had predictive value, because Delta didn't arrive everywhere at the first place at once. But once it arrived, we could call the health department and say, you're going to see a surge in cases in the next 2 to 3 weeks. And it was right. This is kind of the same thing depicted another way. So there was a journalist from Time magazine that wanted to sort of show the effects of the Delta wave. And so what she did is she took all of our sewer shed data and she put it on a common plot. So what this is every line is the number of SARS-CoV-2 or COVID patients in all of these different counties in Missouri. And then based on her sewer shed data, she put them all on where 0 was the time when we first detected Delta. And so you could see there were just no patients in any of them until Delta arrived. Then once Delta arrived, every single case, every single place, you saw these big uptick in the number of cases, which was interesting to see it depicted this way. Interestingly, I asked you to plot it based on vaccination rate to see if we saw a difference in trends depending upon how high the vaccination rate, because Missouri has had a very vast divide in terms of what percentage of the population were vaccinated on a county by county basis, but it didn't really pan out that there was a noticeable difference, at least during the Delta wave, in terms of infections. So then that's what we had until December when, as you all recall, this new variant showed up around Thanksgiving called Omicron, and we were watching diligently for it. And then sure enough, the beginning of December, we first saw it in St. Joseph in Kansas City. We think it probably came in from Nebraska, where they had already had a few cases. It was just a very low percent at first, but it did not stay that way. By the end of December, it was all over the state. Omicron spread in a more predictable way. It spread more like Alpha did. It sort of followed the population centers. So we saw it first, St. Louis, Kansas City, Springfield, and then eventually it was everywhere. So by mid-January, you remember the Omicron wave was intense. So what's been happening since then? So this is the most recent date of our data from our sewer shed. So the yellow line here is the output from wastewater testing, and the little bars are showing the number of actual cases. And so you can see we're sort of in a long, slow wave right now, which I'm calling the Omicron subvariant wave. So it looks kind of uninteresting when you just look at it this way, but when you actually break it down, we've had a number of successive waves. So we had our first wave of BA1, which was this blue line, which was here from December until it finally crashed out in March. And then BA2 moved in, which slowly took over, and it sort of peaked by mid-March, but then it went away too. Then yellow, we had BA2.12.1. I don't know if you've heard of this one, but it was a champion for a couple, about 6 weeks, and now it has finally been outcompeted by what's called BA4 or BA5. Probably mostly it's BA5. You've probably heard about BA5. I'm sure there will be another one down the pipe, but that's where we are right now. I think our wastewater numbers went down finally for the first time since March, but we will see whether that holds. So now to the third and final part of my talk. So I'm telling this differently than I've told it before, but this story will kind of start with an email that I received in about a year ago. It was from a reporter who worked at the Brown Institute. And he said to me, I'd love to talk to you about wastewater surveillance and possible outbreaks in the rat population sometime this week if possible. And this might sound like a completely innocuous email, but this was a 60-minute gotcha moment. He was asking me a question about a research project we had been working on that was completely secret. We had not talked publicly about it, and we were not allowed to talk publicly about it. But somehow he had found out about this and wanted to talk to me about it, and I about shit my pants. I didn't know if I had screwed up. Somehow I had said something to someone that had gotten to a reporter. I was really nervous. But as it turns out, what happened is he was part of this investigatory group that sunshined a whole bunch of health departments in the state of Missouri. And they didn't sunshine my email, but they got a lot of my emails where I had been communicating with various members of the health departments. And there were two stories that they found of mine that were of interest to them. One was the rat one, which I will get into that in a minute. And the other is it turns out they liked my poetry. So this is a weird story. So I had been emailing with the head of the health department in Boone County. And they had this ginormous spike in their wastewater. She had written me and said, what happened with the wastewater this week? She didn't want to wait for the update that they get the next day. And so I wrote back, I can't remember. It was like the numbers tripled or something. And she wrote back, I think she said something like, I think I'm going to be sick. And it was like 5 a.m. and I decided to write her a haiku. And this is a haiku I wrote. Vaccine in a year, but that does not help us to save them from themselves. And the reporters just thought that was really cute and wanted to publish it in their article, so they did. And that's how I'm technically a published poet. But now so the whole rat story, I'll just say that this got some attention. It started out with this reporters. It worked out okay because they found out about a study that was not public, but they in the end agreed to withhold publishing their story, which was the story from the city down in the lower left-hand corner. They agreed to hold off on the story until we submitted our thing for publication, as long as we didn't delay. So this actually put some pressure on us and the health departments to go public with the story I'm about ready to tell you. So yeah, it was in Science. We've had some press in the New York Times, and I about peed myself, but Stephen Colbert at one point even quoted from my article on his opening monologue, which was one of the most exciting things that's ever happened to me. But so here's the thing: is let me remind you, when we get our readout every week, we find out the mutations, and then we look at our cheat sheet to see what variant of concern it is. So here's alpha, beta, gamma, BA1, whatever. So pretty early on, this was March of last year, and this was actually in St. Louis. There was a sewer shed where we were doing our normal analysis, but we started to pull out these sequences that didn't match anything that had ever been seen in patients. And they had lots of mutations compared to what we'd seen before. And when we looked at the mutations, we knew that they were all escape mutations. So we thought we were seeing the beginning of a next wave that just hadn't been seen in patients yet. But then it never showed up in patients. So for 2 months, we were following this lineage from St. Louis that kept changing from week to week, but it never left the sewer shed. It's just stayed in this one sewer shed and then eventually disappeared. And we never figured out where it was coming from, but we decided to name this a cryptic lineage, meaning it was the lineage we were seeing from wastewater that we didn't see in patients. And then we started reaching out to other groups, and in particular, we started working with New York City, and they started seeing wastewater, and they had several cryptic lineages. I mean, there weren't a ton. About 5% of sewer sheds had cryptic lineages, but what was interesting from the ones from New York City, they all had this one particular mutation, which doesn't matter, but it's Q498Y. This particular mutation had never, ever been seen in a patient. However, it had been seen numerous times when people found SARS-CoV-2 strains that became adapted so that they could infect rodents. They looked like, these looked like rat SARS-CoV-2, and that's where we thought at the time that this was coming from. We thought there was an outbreak in the New York City rat population. I'll tell you now, I think it was a red herring. I don't think that's where it was even coming from. But that's what we thought at the time. And we even, we did some experiments. We confirmed that these viruses had gained the ability to infect rat cells. And we also, this is really cool. So we can take wastewater and we can do this thing called ribosomal RNA analysis. So we can find out everything that's contributing to the sewer shed. And this is what we usually find is human and things humans eat. So if you eat a pork chop, there's a good chance we'll see some pork ribosomal RNA. But the one thing we saw that was different in a lot of the New York City sewer sheds is they have a ton of rat feces in their sewer shed, which is coming up. So that was consistent with this coming from, maybe this was coming from rats, but the problem is that it wasn't always there. So some of the sewer sheds that had these cryptic lineages did not have any rat ribosomal RNA. So we didn't know what was going on with that. This is just at the end of, so all through last year, we saw cryptic lineages. During 2021, we were with various collaborators, we were screening about 180 different sewer sheds. And of those 180, there were 9 that at some point had cryptic lineages. And this is just showing where they appeared. There was actually a second one from the St. Louis area that we saw in June of last year that then disappeared for over 6 months and then reappeared this spring and is still hanging out. Haven't figured out where it's coming from, but it's been there. So when Omicron moved in, we decided to step this up a notch. So we could do something interesting, which is because Omicron had so many mutations, we decided to design a screen to see whether there were any sequences that were SARS-CoV-2 but were not Omicron. So we basically designed an assay that would pull out the needle in a haystack. We looked for a way of pulling out sequences that were from an older lineage. And we started reaching out to everyone we could find that would send us their wastewater. And so we ended up getting samples from 38 different states, 7 countries, and started screening them all for cryptic lineages. And the very first hit we got out of this screen was from a colleague of mine in Wisconsin. This is Martin Schaefer. He sent me 8 samples from 8 different communities in Wisconsin, and one of them had this really bizarre cryptic lineage. It was very, very different than anything that had ever been seen in patients, but it was persistent. We saw it all this year. The earliest sample we got was from 2021, but we've seen it every week since the beginning of 2021 until, beginning of 2022 till now. So, this is the detective work we've been doing all year. So we started from the treatment plant that had this, which served nearly 500,000 people. And we started following, so we got all the different sublines which contribute to the main plant, and we checked them all for this cryptic lineage, and we found the one that had it. And then we worked our way up through the city until we got it down to a line, and finally we had narrowed it to a single manhole. So we found a manhole where it could find the cryptic lineage. We could always find it in all of the sewer on the manholes downstream, but upstream it was not there. This is where it was coming in. So we said, of course, what is contributing to this manhole? We figured, what you think, maybe a hospital, maybe I don't know, some kind of place where there's sick people, or maybe an apartment complex. No, it was a factory. This is coming from a place where no one even lives. This is coming from someone who's presumably going to work every day. And so the Wisconsin Health Department basically took over, and they went and they communicated with this factory and said, okay, there's this weird lineage coming from we think from your factory, and we would like to see whether anyone in your factory is sick. And so they said, well, we can do that, but let's first figure out which bathroom it's coming from. So in this factory, they have two different sets of bathrooms, and one of them is only used by their employees, and the other one is used sometimes by the public, some people who come in to pick up stuff. And so we got 24 composite samples from both of these, and only one of them had the cryptic lineage, and it was the one that their employees use. And I just wanted to show that it was by far the highest reading we had ever seen. So this is a log scale. So when we see an outbreak, it's typically maybe 100,000. At the height of the Omicron wave, sewer sheds would maybe have 1 or 2 million copies per liter. The highest number I'd ever seen was from one of the Missouri prison outbreaks, when roughly half of the inmates had COVID. We saw readings of about 50 million copies per liter. The wastewater from this toilet was 10 times higher than that. It was insanely high. So this is really important. So it seems that, in fact, these cryptic lineages are coming from patients. And not only patients, these are probably people that have long COVID and don't even know it. Our best guess is that what's happened is that the virus has moved into their gut, and these people probably think they just have a long-term case of diarrhea. We are yet to confirm that, because we did nasal testing of everyone in the company who was, they explained what was going on to everyone that worked for this factory. Most of them volunteered for voluntary nasal testing, and they were all negative. So we still don't know which person it is, but we do know now that it is indeed coming from a person. So I'm nearing the end. I just want to say that, so cryptic lineages, these are rare, but not exceedingly rare, and they're from long-term infections. So we're starting to wonder, you've heard a lot about COVID, long COVID, and we're starting to wonder whether these might actually be cases where the virus has established itself in some other part of the body. Most people with long COVID are actually negative with nasal swabs for the virus, but it could be that the virus is somewhere else still circulating. So we think they're probably from gut infections. And although these are not an imminent threat, these are a potential threat, because we've actually tested, and these viruses, if they were circulating, would be very resistant to our sort of the immunity we now have against the virus. So I just wanted to share you the teams. So on top, this is the University of Missouri Wastewater Treatment Plant, wearing our uniforms. And then below, this was a few weeks ago, it was really cool. We got an award from the governor, and the whole team with DHSS and DNR and University of Missouri all came together. It was the first time most of us had ever met. And to his credit, the governor shook all of our hands despite knowing what we do for a living, which was surprising. And also collaborators elsewhere. And with that, I would be happy to take any questions.
Wow, that was wonderful. Really, really interesting. I'm wondering if anybody has any questions that they want to ask. Looks like, let's see. No hands up at this point. I don't see any messages. Oh, wait, looks like Norm, you've got to, let's unmute yourself.
All right.
There you go.
Do you get any pressure from politicians who are looking for some of your data to maybe reinforce some preconceived notions they have about the disease, or about where it's occurring, or whatever, some political reason where they might try to gather you as a source?
I'm happy to say no. We have not had ever any pressure to hide our data or of any way. So all of the data goes onto the dashboard as soon as we have it. The one place where I will say is that health departments do not want information that they don't know what to do with. So when we started at our first finding our cryptic lineages, if they don't know that they are causing any health problem, they don't want to cause a panic. So they are very cautious about sharing things that they don't understand. But other than that, no, they have been, we update our dashboard every week, and no, don't get approval from anyone in the government. It just goes up.
Okay, we have a question. You've got to unmute yourself, Donald.
You're muted. Donald, you're still muted.
You're still muted. We can't hear you.
How's that? Now can you hear me?
I can hear you now.
I actually have one comment and one question, or we'll figure out what it is. The first thing is Craig Venter's book, Life Decoded. He mentions that after he had retired, he took his sailboat, which I'm sure was a luxurious sailboat, and started dragging a filter to detect DNA in the seawater. And I'm just wondering if any of those methods were involved in some of the work that you're doing.
So yes, this isn't strictly surveillance, but I am totally into like seeing what's out there. So we have now several times taken samples from lots of places: Missouri River, wildlife reserves, sewer sheds, and then you can do what's called shotgun sequencing. So you just take the RNA and you get rid of all the bacteria and all of that stuff. And basically what you're left with is mostly viral sequence. And then you do what's called shotgun sequencing. So you just randomly sequence little pieces of information, and then you just get an unbiased readout of what's in there. And it's astounding, because like it's crazy. If you take sewer shed, you'll find hundreds of viruses that are completely unknown. They're not even in the database, but they're mostly bacteriophages, but it's actually a little bit, it's very humbling how much of the life out there we don't know about.
I love viruses.
Call viruses, life, whatever. Okay, but I mean, how many viruses are out there that are everywhere, and we just know absolutely nothing about them?
Okay, the question is, you mentioned very briefly about opioids, and I'm wondering, does DEA have any use in detecting fentanyl usage throughout the country?
I think there's a lot of agencies that are starting to take interest in this. I haven't done anything with DEA yet, but I know EPA was inquiring about who had done this kind of thing. Haven't talked to them yet, but there's all kinds of government agencies that seem to have interest in this sort of stuff.
All right. Thank you very much. This is a very interesting discussion. I really enjoyed it. Thank you. I'll mute myself back.
Thank you. I think Fran has a question. Fran, go ahead.
Thank you. I just wondering if you could walk us through how to get to that information every week.
Oh, sure. Let's see here. No. Do I have? Actually, Laura, if I send you the link, can you post it in the chat?
Yes, or the other thing is I can send it, anything you want, follow up to everyone who's part of the program. Whatever is easier.
Well, it's pretty simple. No, that's not the one. I don't, nope, not that one. Here we go. I just sent it to you. It's a very short, it's just too long to say, but it's, did you get it?
Not yet.
Okay. It's,
I have not gotten it.
it's health.mo.gov/mo wastewater COVID. So it's kind of long, but it's not that long.
Okay. We do have another question.
Shoot.
From Carlotta in the chat room. Do you think that the cryptic lineages are not seen in patient samples due to lower coverage and depth in sequencing swab samples?
Probably not. The thing is with the cryptic lineages is that we've the record there's one that we've been seeing since January 2021. So what is that? 19 months. But we've only seen it from one sewer shed. So if it were actually a virus that we're spreading, I mean, even if it's spreading at low levels, people, I mean, if I've learned anything from our studies of the SARS spread of this virus, is that we humans, we're filthy little creatures. We don't stay put. I mean, when you look at how quickly like Delta and Omicron just move right through the state, it doesn't make sense to me that a virus would stay geographically constrained. And because even if it's like 3 or 4 people infected, you would expect them at some point to go, give it to their cousin across the river or something else. A lot of them from New York City, like the sewer sheds are one is literally across the street from another. So the fact that it remains geographically constrained means to me it's either in a animal population that cannot leave that sewer shed, or it's probably from a single person. So I don't think it's just that we're missing it. Although, I mean, even if it's a person, it is a person out there somewhere. I don't know if they would even test positive with a nasal swab or not, but I know that they are shedding ungodly amounts of virus into the sewer shed every week. It's quite amazing.
There was also a comment from Keith that talked about extending this technique to other viruses, for example, polio, which, it's been in the news lately. Are some pathogens more or less or not at all detectable by their nature? So that's the question.
I've got more press about looking for monkeypox, and I haven't even seen it from wastewater. There's I was skeptical at first whether you could detect all different kinds of viruses. Like I was very skeptical about flu, but people have actually had really good luck with that as well. So sky's the limit. Pretty much. I mean, it might be really. I have my doubts that we'll be able to detect HIV efficiently this way because it's in the blood. It doesn't really end up in your feces, but anything respiratory or gut or GI, I think there's a very good chance we would be able to detect it.
The one thing that I thought was interesting is I've heard of watersheds before, but I actually have never heard the term sewer shed before. So
it makes sense though, right? It's just all of everything that contributes to that.
But my question is how big are sewer sheds? I mean, is it for instance, here in St. Louis, is it the entire region, or are the sewer sheds smaller?
The smallest sewer shed we have is a few hundred people. The largest one is a few million people.
Wow. Okay.
Our cases of COVID going up or down right now, they seem to be going down slowly. I'm sorry if you wonder. COVID cases. So we've been with this Omicron sort of, I call the subvariant wave. It's been like a slow motion surge, but I think we're just cresting and it's starting to go down again. I mean, the good news is that the hospitalizations have not been as high as with other variants, particularly now. But I don't know about you, but everyone I know has gotten COVID in the last 2 months and has managed to avoid it up until now, including me. So I turned 50 in May. I got my 4th vaccine, and a week later, I finally got infected. So a lot of people are getting infected right now, but it seems like it's finally started to go down. I think I've said that before, and then it's surged again, but I think this time it's starting to go down.
Do you see the question from Denise?
Yeah. So the thing is, yes, we want to do this, but it's not a thing. It's not, there's no FDA fecal test for COVID. There is no sort of, there's no known GI disease that's caused by COVID. So if we want to do this, the way the health department feels about this is that they think this is interesting. They think this should be followed up on, but they do not think this is public health. They think of this as research. So if we want to do fecal testing, we have to get what's called an IRB. So a way of where we get permission to get patient samples for the use of research, and then we will be able to do the screen. But we can't do it yet. If it started spreading again, that would be a thing. But right now, as far as we can tell in Wisconsin, this is one patient. And we don't even know for sure whether it's really even hurting them. It could be that it's not actually causing them any harm. So they don't want to, it's sort of a do no harm kind of thing. And until they know something is a problem, they don't want to try and fix it. That's not true. They want to fix it, but there's certain boundaries they can't cross.
I think Norm must have another question. Go ahead.
How many people on your staff at Mizzou?
So mine is actually mostly undergrads. So I have 2 full-time people, and I have 2 graduate students who are sort of peripherally involved, but I'm mostly doing I have like 8 or 10 undergrads that do most of the work. Chung Ho is the lab is a little bit bigger. He has about 6 people. So collectively, we'll probably have about 20 people. But like I said, most of them are undergrads that are part-time.
Fran has a question. Do you sterilize equipment between testing?
We usually, it's disposable. So the tubes come in and then we throw them away, and then yeah.
So that was actually sort of my question, is who's actually getting the samples? Because you just talked about the tubes. Who's actually getting the
Oh, those are the actual wastewater operators, and those are samples they were collecting anyway. So they collect wastewater samples to look at things like total solids and various other things. This is something they were doing anyway. All we're doing is having them take a little bit of what they're already collecting and putting it into a tube that they ship to us. So a point I should have made, I'll make it now. We have not been able to culture virus from wastewater, and believe me, we have tried. The virus that's in wastewater does not seem to be infectious. I'm not sure why it isn't infectious, but even that, even that sample that had 500 million copies per liter, both at the University of Wisconsin and here at University of Missouri, we tried to get virus to culture out of it and we couldn't. So wastewater is something you need to be careful about because there are a lot of nasty pathogens in there, but COVID is not really one of them. It's not a source of infection.
That's really interesting. There's another question. Are samples shipped on cold chain like we do for polio? I don't know what that means. You might want to explain what that means for everybody.
So a cold chain means you keep it cold. Some things like, so the mRNA vaccines you get actually have to be kept frozen. I remember that was a big deal with Pfizer,
Right.
that it had to be at -80 until you only thaw it once and then use it. With our wastewater, interestingly, you can't freeze it. If you freeze it, it kills the signal. So it has to be kept, it cannot be frozen, but it's actually remarkably stable. We do ship it with ice packs to keep it cool, but it's not actually even really necessary. It's the signal is remarkably stable. We'll have wastewater in the fridge for months and we can go back and still detect it with no problem. I'm sure it has a half-life, but it's a very long one. But as long as you don't freeze it, it seems to last for a really long time.
Okay. Does anybody else have any questions for Dr. Johnson? Because this has been really interesting. Okay. I think Fran is clapping. So we are very, and so is Norm. We're very appreciative of your time and your expertise. And, depending upon how this unfolds, maybe we'll be able to have you come back and give us an update.
All right. That sounds great.
Okay. Thank you so very much. And thank you everyone for joining us.
All right. Bye-bye, everyone.
Bye-bye.